GIGA Platforms, and at IRFAS, the Technology Transfer Office of the University of Liège. I'm very pleased to welcome you today for the first webinar of our series GIGA Platforms, Key Actor for Your R&D. So our objective is to let you know and give you facilitated access to the multidisciplinary expertise of GIGA platforms, the state-of-the-art technology, the advanced equipment, and also to emphasize the potential collaborations with your companies or your academic lab according to your needs. So today's first topic, platform proteomics. We'll speak about mass spectrometry and protein characterization with application in R&D and quality control. So we'll spend 1 hour together with our keynote speakers until 12:30. Afterwards, from a 12:45, you will have on demand the possibility to meet privately our researchers in speed dating today or later on. You just have to ask Aurélie, which is in the back office. You send her a private message with the chat conversation tool. And if you have any question, I suggest that you also use the chat conversation tool to announce us the subject or main topic of your question. And in the back office, we have Aurélie and Maximilien that will gather the questions for the end of the public sessions. So before giving the word to the first speaker, if it's okay for you, I invite you to cut off your microphone during the presentation. We will begin showing you a 2-minute video of the whole concept of the GIGA and therefore the context of the proteomics platform. So I'll ask my colleague Florence. Florence, could you please launch the video and share your screen? About the complete ecosystem at the service of academic researchers as well as companies, I have the pleasure now to welcome our first speaker, Karine Bebron, who is the manager of the technological platforms at GIGA Research Center. So as background, Karine has a master in biochemistry with PhD in science. She's graduated from the University of Liège, postdoc in Liège and Aachen in molecular biotechnology. Karine has a mixed career in academic research and in industry, as she has worked for Symbiose Biomaterials, a technological innovation platform very close to GIGA and close to the steel industry. She has also worked in chemical industry as an R&D and procurement managers. And 2 years ago, she went back to GIGA as manager of the technological platforms. So Karine, your microphone is on, your camera also. Please share your screen and we're listening to you.
Good morning. First of all, I would like to thank you to attend, and I hope that you will enjoy to learn about our proteomics platform, and that will give you the desire to work with us. You have just seen a short presentation film about GIGA, but let me briefly highlight a few points. GIGA is University of Liège Interdisciplinary Research Institute in the biomedical sciences. The 600 members of GIGA strive for academic excellence to foster groundbreaking medical innovations. The location of GIGA within the University Hospital of Liège allows a constant dialogue between researchers and clinicians, and thus promote the development of efficient translational research. The GIGA is composed of 10 thematic research units revolving around 4 main axes: cancer, cardiovascular diseases, all the diseases related to immunity, inflammation, and infection, and the neurosciences. Technological platform results from a pooling of a set of technological and human resources in order to offer cutting-edge technology in line with the needs of researchers from academic and industrial worlds. GIGA supports emerging startups and spin-off in its lab hotel incubator. The lab hotel is an innovative concept of business facility. It's an industry-dedicated lab, fully integrated into the GIGA Research Open Lab, and companies have a turnkey solution with an equipped lab, a full access to the shared equipment of GIGA, and to the technology platforms. Thanks to all of these elements, GIGA is an ecosystem promoting the development of the pharma, the biopharma, and biomedical sector. The ability to explore the genome, the transcriptome, and the proteome of different organisms, to systematically analyze the generated data thanks to bioinformatics, and to place them into a cellular or organism context appear essential for any research or active company in the biopharmaceutical, biotechnology, or even agri-food field. However, access to exceptional technological equipment for research and innovation is becoming difficult, especially for SMEs, given the cost of implementation and the growing expertise that they require. The objective of the GIGA technological platforms is thus to meet the needs of the biomedical sector by providing quality technical support and cutting-edge expertise to industrial and academic research and innovation activities, from basic to preclinical and clinical studies. GIGA technological platform bring together advanced technologies, exceptional equipment, and high-level skills in the following 8 areas. Next to proteomics, which is the topics of today's presentations, zebrafish and flow cytometry and genomic bioinformatics platforms will be the subject of the next webinar of this first series. You will be reminded of the dates at the end of this seminar. But the GIGA platform are also preclinical and clinical in vivo imaging, viral vector, immunohistochemistry, and animal mice facilities. These 8 platforms are accessible to researchers from all academic and private sectors. For example, we work with 45 research teams out of Liège, but also with 120 companies, SME, from Belgium and abroad. In order to stay at the forefront of innovation, to meet the challenges offered by companies, both technological and in terms of quality standards and speed of executions, to further improve the services provided to researchers, and finally, to ensure that they are in suitable infrastructures, GIGA platform are first continuously completing or upgrading the current equipment, and secondly, providing the expertise required for the provision of services on this advanced equipment. The platforms employ 30 logisticians and technician specialists in the field. You can find more information about each platform on our website. And please don't hesitate to contact me for more information or to start a new collaboration. Thank you.
Thank you, Karine. Let's now focus on proteomics, the topic of our today webinar. So therefore, I'm very pleased to welcome Dominique Beweer, who is the manager of the GIGA Proteomics platform. Dominique is a chemist. Her expertise field are mass spectrometry, proteomics, analytical chemistry. She has previously worked 7 years in the mass spectrometry laboratory and also at Cienzano, the Belgium Scientific Institute of Public Health. And since 2011, she works in the GIGA Proteomics platform, and she has acquired expertise in protein analysis and proteomic analysis. She's the manager of the GIGA Proteomics platform since 8 years now, working in close collaboration, of course, with the mass spectrometry laboratory, so especially with Gabriel, that you will hear a bit later. And Dominique is also manager of foliage at the MS Quanta platform, an inter-university platform, Liège and Mons, dedicated to the validation of new biomarkers. So Dominique, tell us more about the available services of the platform and their application in R&D and quality control. And maybe while she's preparing a screen before sharing it with us, I remind you not to hesitate to use the chat conversation tool. Let us know the topic of your question or to ask Aurélie a private meeting with our expert. Aurélie and Maximilien are welcoming your message in the back office. So if Dominique is ready, the word is yours.
Thank you, Fabienne, and thank you to all of you to attend this first webinar of GIGA Technological Platform. So today I would like to take the opportunity to present you some of the services the GIGA Proteomic platform can offer to characterize biomolecules. And so the outline of this presentation will be a very brief introduction over the platform, and then a description of the services we can offer from small molecule characterization, protein and peptide analysis, but also a post-translational modification characterization on protein. And all these analyses can also be used to characterize raw material containing protein samples. The last services I would like to present you will be quantitative differential proteomic using level 3. And I will finish by presenting you the MS Quanta platform that Fabienne just told you about between University of Mons and University of Liège. So as an introduction, as we say, the GIGA Proteomics platform is working in very close collaboration with mass spectrometry, a laboratory of Professor Gauthier Epp in Liège, because most of our services are based on mass spectrometry with or without a separation with chromatographic media before. We mainly use a liquid chromatography as peptide and protein are quite polar compound, and we have a broad range of UPLC available, two UPLC with what is called normal flow rate, and 3 nano UPLC, which 2 of them are 2D nano UPLC with 2 reverse phase separation. For the mass spectrometer, the instrument are shared with the mass spec lab, and mass spectrometer has 2 main characteristics: the source, which will help to bring molecules from the liquid to the gas phase, and the analyzer that will separate the ion in function of the mass to charge ratio. So for the mass spec, we have access to a bright set of mass specs starting from low resolution like a quadrupole. Here it's a triple quadrupole, mainly used for quantitative analysis I will describe later. Also ion traps with special fragmentation capabilities for mainly glycopeptides or glycoproteins. A range of time of flight mid-resolution mass spectrometer that are really versatile spectrometer using proteomics. 2 orbit traps that are using mainly for differential proteomics, but also for sequencing of pure protein, and very high resolution mass spectrometer. It's an FT-ICR for characterization with a very precise isotopic distribution. Most of these spectrometers are equipped with an electrospray source, which allows to bring ion from liquid to gas phase for the protein in the ion without breaking them. But we have also 2 spectrometers, the MALDI-TOF and the FT-ICR, that are able to ionize with what is called MALDI. In these analyses, the proteins are co-crystallized with a matrix which will absorb the UV radiation of a laser to allow the protein to come in the gas phase. So, which services can we offer you? First, with the FT-ICR, as I told you, it's a really high resolution mass spectrometer that allows to determine molecular formula for small molecules, molecules that have a mass below 1000 Dalton, and it allows to give you the molecular composition of your molecule. So is my molecule correctly sensitized? Or if you have a gas for a purified compound that you have isolated, is what I was expecting. And if we combine the simple MS spectra with tandem MS spectra, which means we will fragment the molecules in gas phase, you can gain more structural information such as branching of the compound or the differentiation of isomers. But let's go back to the core of our business, if we can say, the protein and peptides characterization. First services I would like to show you, it's the mass determination. That's what the mass spectrometry can make at simple, giving a mass. Here, the first thing I will present you is with the MALDI. So we co-crystallize the protein with a matrix, and then we send them to gas phase and analyze with a time of flight mass spectrometer. And we will get this kind of spectra, which is quite simple to interpret because MALDI has the properties to mainly generate single charge ions. The spectra you are seeing here are antibodies, And you can see that we have a difference between the two spectra. And what is this? This is an antibody that has been, conjugate with aptan. And if we look closer to the singular charge peak, we can see that the apex of the two compounds are different, and there is a mass shift between these two apex, and this mass shift, the difference of mass between the native antibody and the conjugate antibody, can give us an average number of conjugate that has been covalently linked to the antibody. The other way to determine the entire protein mass is using electrospray, still with a QTOF analyzer. And what is the difference? By bringing the protein in gas phase using electrospray, we will generate multiple charge states. This will lead to a more complex spectra that we can see here, with more peaks present for one protein. The spectra, which is shown here on the left, is a mix of two proteins. This is a mix we use as a QC, a mix between these two proteins that when you process the data to what is called deconvolute and to get all the charge state of one protein bring to a single peak, we can see that we get one protein around 12,000 Dalton, and the BSA, which is our second protein, which has a mass around 66 kilodalton. But these techniques can also give us an insight between the forms that are present in a protein. Here you can see an example also of an antibody. On the upper spectra, this is only the heavy chain because we have reduced the antibody. And so we are looking at the heavy chain of the antibody, and we can see that there is mass shift between several forms of the antibody, and the mass shift can correspond to one exose in this case. So we are looking at the different glycoforms of the antibody. This is quite a difference between the MALDI spectra, because we had an average of the distribution of the form that were present in the protein. With the electrospray, we can really look at each form separately. The drawback is that we need more sample to do the analysis, and that usually electrospray is suitable for proteins that are at maximum 70 kilodaltons, despite MALDI can goes up to 200 kilodalton. So we have the mass of the protein, but usually it's not enough to be sure that we have produced the right protein. We know, okay, our protein has the expected masses, but is the primary sequence of the amino acid really correct? To know this, we have to sequence the protein, and this can also be done using mass spectrometry. To do this, we will first digest the protein using enzyme, usually trypsin, to generate peptides. Those peptides will be separated using LC-MS, and we will have a chromatogram with the different peptide of the protein. We will then select each peptide sequentially to fragment the peptide and obtain the MS-MS spectra of a peptide with different amino acids sequence done, which will allows us to really confirm the amino acid sequence of the peptides. After, the peptide sequencing will be aligned against the theoretical sequence of the protein. So this is what you usually get when we do trypsin digestion of a protein. Trypsin cleaves after lysine and arginine. So for a given sequence represented here in red, you will have all the blue peptide that will be generated if you have a complete digestion using trypsin. The problem is that you do not have access to all the peptides in LC-MS analysis, because some peptides are too short and so are not retained on the C18 LC, LC system, and some will be too long and then they will not be fragmented enough to be really sequenced and so to be identified by the bioinformatic pipeline. So if we look at the really suitable peptide for LC-MS analysis, we only keep those peptides, which you see doesn't cover all the sequence of the protein. So we can still say, okay, this is protein A, but we cannot have information of the sequence, for example, for this part of the protein. So in collaboration with the mass spectrometry laboratory, the platform and the omic part of MS Lab develop a multi-enzymatic limited in-time digestion, which is called MELD, which will use several enzymes at the same time, but during a short time to produce more peptides and more different peptides. As we do limited in-time digestion, some cleavage site won't be cleaved during the 2 hours of reaction, and so we can generate different peptides. Still, there will be peptide that will not be suitable for the LC-MS analysis, but when we do the same as for the trypsin digestion, we keep more different peptide than for trypsin. And what is really interesting is that we have several peptides covering the same part of the sequence of the protein. And as we have a diversity of peptide, we are able in general to cover 100% of the primary sequence of the protein. And you can also, using these techniques, having an idea of the PTM that are present on your protein, given that you know which PTM you are looking at. So the MELD is really a powerful techniques to identify protein, but also to do an extensive characterization of the sequence of your proteins. And Gabriel will speak after of even more development that are going to bring more PTM characterization using these techniques. So that was for the sequencing. Now the differential proteomics, this is quite the same because we will still identify protein, but we will add the quantification of the protein. Not an absolute quantification, but a relative one that will allow us to compare 2 or more conditions. So if we have a condition A and a condition B, we will digest each sample and then subject them to LC-MS analysis for protein identification. We will have the identification of the protein, but also the abundance of the protein due to their peptide that will be identified. And as you can see, you can have some peptides and so some protein that are not present at the same amount between your 2 conditions. So the results you get from this type of analysis is a list of the protein identified and quantified in each sample, and you can calculate the relative amount between condition A and B, and doing statistical analysis to find the relatively differently abundant protein between conditions. One thing to mention is that this analysis, as we have a mix of peptides, will be highly dependent of the dynamic range of concentration of protein you have, such as in sample like plasma, when you have highly abundant protein like HSA and IgG, we will have lower number of protein identified, up to 300, but usually with a cell extract or tissue extract, we will get from 1,500 to more than 4,000 protein identified and quantified in each samples. One drawback of this analysis is that as it's a complex mixture, usually we use 2D UPLC, so the analyses are quite time consuming because it takes nearly a half day per sample of analysis. And as to get the differentially abundant protein between condition, this relies on statistical analysis. We need at least, the very least, 3 biological replicates per condition you want to compare. This method has been further developed to can work with less starting material, because what I just showed you before is usually starting from 20 micrograms of protein. And sometimes we do not have this quantity of protein, or we want to take advantage of really localized proteomic analysis. This can be done with a method developed also at the MS Lab that is now available on the platform, that coupled the laser microdissection available on the GIGA imaging platform to the discovery proteomic I just showed you. So we can, starting from less than 3,000 cells, identified more than 2,000 protein per sample. One example of these techniques is one made with the team of Michael Erfs from the GIGA, who studied 2 zones of development of cancer, one squamous and one transitional zone. He laser capture microdissect a section of his histological slice, and we did the label-free quantification. So here is an example of the result you can get from such analysis. First, we have the classical Venn diagram, when you can see that nearly 3,000 proteins were identified that were common between the 2 zones of dissection, and you have the identification of the protein with the difference of abundance between the 2 zones. The vast majority of the protein are not modified. This is the hypothesis of label-free proteomic. The vast majority of protein doesn't change between conditions, and only a small portion is modified by the change of condition. And these are the proteins you can see here highlighted in red. And these proteins usually are considered as biomarker that will be validated later using usually immunochemistry, also available in the GIGA platform immunochemistry. And you can also do with those results, having the protein identify enrichment test to see if a specific pathway or a specific biological process is implied between your condition. So as I told you, label-free proteomic is usually used for the discovery of the biomarker. Then those biomarkers will be verified and validated, usually still by mass spectrometry, but using not more discovery, but targeted mass spectrometry I will just explain after. And after, the main validation is done on more than 1,000 samples using ELISA test or immunochemistry test. So the validation of the biomarker are usually done using targeted proteomic, which is done on triple quadruple mass spectrometer that we allows us to select in the first quadrupole only the peptide of interest we want to follow for the protein we know will be different between our condition. And we will fragment the peptide in the second quadrupole and select the fragment of interest, those who are specific to the peptide, in the last quadrupole. This will allow to focus only on what we want to follow, and so to increase the sensitivity of the methods. And you can even, using this technique, do absolute quantification if you have what is called usually heavy peptide. This is a stable isotope label peptide with same sequence as the endogenous, or even better, the heavy protein. We have recently finished validation of biomarker with the team of Edouard Louis, when we followed more than 70 protein in plasma over more than 100 samples in only one campaign. Last thing I would like to share with you is the MS Conta platform. MS Conta platform, as Fabienne told you, is an inter-university platform between University of Mons, ULB, and ULiège. For ULiège, this is the GIGA Proteomics facility that brings its expertise and its instrument. And in University of Mons, this is Proteomics and Microbiology Laboratory of Professor Rudy Wattier that brings also its expertise and instrument. What is the goal of this platform? It's to reach really a critical size to can provide you a high throughput analysis for biomarker validation, so for quantitative, but also qualitative analysis. And this facility is open to every partner, even academic or companies. So I hope that I could show you with this short presentation that mass spectrometry is a versatile tool and that it's really useful to characterize protein and peptides. Thank you.
Thank you, Dominique, for your explanation. So as quality control plays, of course, a critical role also for the biomedical laboratories, we have the pleasure now to welcome Joël Ouidard, who is the quality manager of the pharmacy department here at the University of Liège and the quality manager of the laboratory of mass spectrometry, both certified and accredited. Joël is civil engineer in chemistry. Her expertise is the quality management for different applications, analytical laboratory, quality control for drugs and for food. She is also technical and quality system auditor for BELAC, the Belgian accreditation, concerning the main accreditations for quality management in biomed labs. So Joël has accepted to share with us her expertise and experience in this matter. So please, the word is yours, Joël.
So hi everybody. I will show you some stuff about quality at GIGA Proteomics platform. Sorry, first of all, I would like to show you just some referentials that are turning around our subjects. It's medical devices, it's GMP, GLP, GCP, and the ISOs. If we take, for example, the drugs, from a molecule that can be interesting to a drug, there are some different steps. The two first has no rules or not yet any rules. It's the research part and the drug discovery. If you want to go the next step, that would be the toxicological study, so non-clinical study on animals, you have to be GLP certified. For the human testing, it's clinical trials, you have to be GCP certified. After that, you can ask your market authorization to be allowed to manufacture the drugs, and you have to be GMP certified. The last part is the GDP, is only for distribution and storage of drugs. After this short introduction, I want to show you some examples of quality in a research and services laboratory, such as GIGA Proteomics platform. For that, I will use the 5M method to go through each items. The first one is the man. So in the laboratory, all the analysts are trained to be able to do some steps of the analysis. You have here as an example of a record of a training. So Nancy trained to be able to digest proteins in solution or gels, and she also trained on the using of a mass spec, and she followed each steps to read the procedures, observe another analyst, and after that doing the step of the analysis. All those are registered, and you have a function sheet that collects all the abilities of Nancy. So she's, for example, qualified to use and maintenance the QTOF, but she's not yet formed to use and maintenance the FTMS. All those papers can allow the managers to attribute some analysis to one analyst or another, knowing which kind of analysis they are able to do. About the methods, we have to discuss with you the objective of the methods. As Dominique said, we have quantitative, qualitative method. Depending of the objective, what are your objective? We will propose you the electrospray or the MALDI, depending of what you want to do. During the steps of development of the method, we will ask us, is the method able to answer those objectives? We will try also to have an idea of the robustness of the method for your specific applications. And during the time, we will ask us, is it still appropriate? Sample types is the same than in the beginning, and so on. We will check everything about the method. All those methods are, sorry. Oh. All the methods are written on some standard operating procedures, so SOP. You have here the example of the first page of the digestion procedure. So each steps are explained, and you have all the steps that are registered. So the analysis write down every steps they made, all the batch numbers of the solvents, everything that is used, which equipment was used, what are the parameters in the database searches, and so on. Everything is written and is checked at the end of the analysis. And those information are also registered in the report that you get. So the report will look like this. You will have the information about the device that was used, the experimental conditions, and one point that is really important is the quality control sample that we put in parallel of all the samples. So you will have which kind of quality samples, and you will have also the result of this quality control. This quality control is also analyzed for the trends. We check this from analysis to analysis, from day to day, and we try to optimize the things. We check if everything is good. We try to prevent any problems during the procedure. Concerning the milieu, we have some rooms that are restricted to the access. We also have freezers and fridge that are restricted with keys. One big point is the machine. So all the mass spec and all the equipment are covered in the quality system. They have some user's manual, so everything is written down. And we also have some notebooks where we write what is done on the machine in terms of samples, quality control, but also repairing, maintenance, and so on are all registered. Our equipment are in an inventory, and there is a sticker here. You can see that it is green, so the mass spec is in order of use. All the equipment that have to be calibrated are also identifying that they are calibrated in due time. Do not pay attention to the date. It's quite an old photo, so I see that it's 2016, but they are now also calibrated in this year. The fridge and freezer are also followed for the temperature. We note everything and we act if there is something wrong. For the machines, we also check the performance of everything. We have several indicators that can help us to prevent some problems, like in parallel with the quality control. Here it's a MPDS mix, sorry. It's a mix of 4 digested proteins, and we check if after the analysis we can recover all the 4 proteins, which is what is the score, the number of peptides and the sequence coverage. And if the indicator are lower than our specification, we do something. Here you see that there are 3 levels that are not satisfying, so we made a maintenance before analysis another sample. We have some other example of indicators. Here it's a pressure indicator for the LC, and we also have the accumulation time for the mass spec that can inform us about the performance. The last M is the materials. If we have to, so for example, to prepare some prepared solution, we indicate the temperature storage, the label, the identity of the solution. We also put the preparation date, the expiry date, and the name of the person who prepared it. And there are also some preparation sheet where we register batch number and so on. Here are the 5 items that I could be able to show you easily, but there are a lot of other stuff that are managed in the quality system of the lab. We manage complaints, we manage contracts, management review are organized annually. Impartiality and confidentiality are also quite hot stuff for the moment. The records, you see some example of records, but there are a list of records also. Data integrity is quite a hot subject for the moment also. We do deviation, we register the deviation, we act with the corrective action. We do risk analysis about quality and security for the lab. And we have also internal audit that check that everything is in the good way. What about our external recognition? For the moment, we do not have any official external recognition because we had an agreement of the agency till April 2019, but as the law changed, we had to stop this agreement because it was not affordable for us at this moment. But till this moment, we still have some customer audits. So our customer comes and check if everything is good. And those customers are still working with us, so it's not so bad. But during the last management review, we defined the objective to get the accreditation for this part of the lab, and we choose the ISO 17025, and the objective is 2020 to get this accreditation. We can also work with the GMP because the pharmacy department is certified for GMP, and we can pass through the pharmacy department to do the analysis for GMP for raw materials or finished products. I give you a lot of information about quality that is probably not your specific tasks. So if you have any question about regulatory affairs and so on, do not hesitate to contact me. You have here my contact information, or you can go from by the GIGA platform also to contact me.
Thank you, Joël. So the main support of the proteomic platforms is, of course, the advances in science. And we wouldn't like to leave you without giving you an overview about the ongoing developments for advanced biomolecules characterization. So therefore, I'm very pleased to welcome Gabriel Mazzucchelli. Gabriel is associate professor. He is the scientific manager of the mass spectrometry laboratory of the University of Liège, in which is in charge of scientific collaborations concerning developments in mass spectrometry for the so-called omics application. So its research projects focus on the characterization of proteins and proteomes in biological systems using advanced mass spectrometry. In 2008, Gabriel became scientific and QC manager of the GIGA Proteomics platform and of the Omics academic research unit of the mass spectrometry lab. He became associate professor in 2015 and is currently a vice president of the Belgian Proteomics Association, and is involved in international organizations as leader of proteomics working groups. So Gabriel, if we are ready, we are listening to you.
I am. Thank you, Fabienne. Hello, everyone. So today, during this very short presentation, I would like to show you the ongoing developments at the MS Lab for Advanced Biomolecule Characterization that will lead to new services through the GIGA Proteomics platform. So I will focus my talk on the deep protein characterization, but I would like to mention that we are active in MALDI MS molecular imaging. So in that technique, we use MALDI to localize molecule on tissue slices directly. We are also quite active in downscaling MS analysis. So the goal here is to lower the detection limit to go to single cell proteomics analysis, and we have been granted recently for a new project of more than 2 million euro with several groups of Liège, but also with Ciris and Diagenode, and we will try to create a new microfluidic chip to process the sample prep for such kind of analysis. And the last point is that we have a joint effort with the pharmacy department and Pascal Di Tullio to create a new platform, metabolomic platform with NMR and MS analysis. So if you have some questions about these three last points, please do not hesitate to contact us back after the presentation. So let's go to the deep protein characterization. When a company produces a protein, of course, you have to check for the identity, but also for the folding of the protein. I will not go into details about the folding, but just mention that we have now a new system, an hydrogen deuterium exchange coupled to mass spec, which is under development. And in the near future, we will be able to propose epitope mapping experiment. About the identity of the protein, when you produce a protein of interest, of course, you want to have only that one, but it's not always the case because you co-produce undesired forms of the protein. And these can be post-translational modifications, clipping forms, mutated forms, but also chemical modifications. So you can have quite a huge diversity of co-produced forms and with different amounts of them. So then comes the nightmare and all these questions. How many forms do I have? What are their relative abundances? Which modifications do I have? And where are they localized? To finally ask the final question, why do I have this? Of course, to understand why you co-produced undesired form, you need first to answer these first four questions. And here we would like to propose our strategy, which is composed of the combination at least of two methods, one related to the anti-protein analysis and the other one related to the protein sequencing. For the anti-protein analysis, when you have such kind of mixture, you can apply 1D gel, 2D gel. If you want to have more resolution, you have also LC-MS or UV analysis or direct MS analysis. And with these approaches, you can look at how many forms you have. You can perform the relative quantification, but in general, you have a low separation pore and a low dynamic range. And also you have no qualitative information. It is why you need to go for protein sequencing. So what we propose is to use our MELD approach, so multi-enzymatic limited digestion, to generate a lot of different peptides. And thanks to that, we are able to detect a lot of modification and to localize them. Of course, you lose the peptide protein linked because you digested them. And then you have not the possibility to relatively quantify the different forms. It is why you need both approaches. You can combine also both by directly, for example, here, collect a fraction of interest and process to the same workflow. So this is just an example of what you can get if you apply our MELD approach on the human serum albumin. Here you can see the sequence of the protein, the beginning of the sequence. Each blue bar corresponds to a peptide that matches the sequence. And this is the rest of the sequence of the HSA with only one analysis. And as you can see, we generate a lot of unique peptide, and we were able to cover 100% of the sequence with more than 1,000 unique peptide with very stringent parameters. So the false discovery rate is lower than 0.1%, and the error in mass is lower than 3 ppm. So with that, you can look for non-modifications. Here is an example for glycosylation. And as you can see, all the peptides, the different peptides here that has been detected bear the same glycosylation modification. And this is true for all other PTM. At the end, you can have really an overview, a global overview of your protein sequence and post-translational modifications. Now, if we want to go a little bit further, we can combine the MELD approach with supercomputing approach. This is quite interesting because you can apply an untargeted PTM identification. How does it work? The system is first identifying the signals and try to identify, as usually, the peptides. And based on that, the system is building a new database of identified targets. And then the system is looking at all unidentified features and try to match this unidentified feature with the previously identified target if we add a delta of mass. And the system is looking at all different delta masses, but also it's trying to localize, thanks to the MSMS spectra, where this delta mass can be associated with. So in final, you can process all the unmatched features. We have an unlimited number of modifications. We can localize these modifications, and thanks to the MELD, we have an increase in efficacy. So you can have such kind of graph where you have an axis here in the x-axis all the delta masses that were detected, and here the frequency, the number of detection of these delta masses. And in that example, you can see that oxidation has been seen 100 times. You can, of course, know what are the peptides that are behind this modification. And you can have also some that are not explained by the database, but you know the delta mass, so you can make some hypotheses. So with that, because I'm already out of time, I would like to stop and thank you for your attention. And if you have any questions, please do not hesitate. Thank you.
Thank you, Gabriele. So indeed, does anybody has some question? If you have, don't hesitate to raise your hand. So with the reaction tool on Zoom, or if I don't see you, don't hear you, just put on your microphone, your camera. Annick has a question, I see. Please, Annick.
Yes, so Annick Ubrex from the Interface. So being a former protein scientist, I was just wondering if it's possible to determine the entire protein mass just starting from a SDS page. Can you just clarify, please? Thank you.
This is a question for you, Dominique, if you want.
Yes, just putting my microphone on. Yes, thank you, Annick. Unfortunately, not, because when the protein are in a page gel, unfortunately, we cannot extract them when they are entire from the gel. You can only extract the peptide from the gel. So no, for an entire mass protein determination, we have to start from a solution.
Thank you again. I wish you a nice day and also success in your own projects. And we are really at your disposal. So don't hesitate to contact our experts or the organization. Thank you very much to everybody.